Hello everybody, I'm Grace Vinodhraj Arulraja from Neshe, Sri Lanka, currently studying Bioprocess Informatics at the University of Weinstefan, Triesdorf in Bayern, Germany, and I have a basic background in medical biology from the University of Duisburg-Essen. As a final project of the program Bioinformatics for Infectious Diseases by PineBio, I have chosen the topic Human Bronchial Organoids as a 3D in vitro test model for SARS coronavirus 2. Today I would like to present our project and the finding from our study, the influence of different human reference genome in RNA sequence analysis and their impact on an in vitro COVID-19 drug test with chemostat. It is a joint project with Mr. Priyankan Sellachami, also known as Victor from our course. Even though my partner prepared some interesting slides about the biological background regarding SARS coronavirus 2, he couldn't able to present his part of the project since he got busy with his thesis works. So let me cover the theoretical part of our project briefly and let me get into the technical part which I was mainly involved in. We all know about the impact of SARS coronavirus 2 around this world, and we discussed enough about spike proteins and ACE2 receptor in our cell membrane. In our project, we observed TMPRSS2, which is transmembrane serine protease subtype 2, which play an important role in activating the spike proteins according to the scientist of the German Primate Center, also known as Deutsches Primatensyndrom. Once the spike proteins get activated, the virion could attach to our ACE2 receptors in cell membrane. To avoid the activation of spike protein, we are going to study about TMPRSS2 and chemostat, which is also known as TMPRSS2 inhibitors. Chemostat is an approved drug in Japan which is widely used for pancreatic inflammation. As an innovative attempt by a Japanese research institute of microbial diseases, they generated human bronchial organoids out of human bronchial epithelial cells. Furthermore, they applied it as a test model for their studies. Moreover, in human bronchial organoids, they were able to observe different cell types which are significant for their studies as well as histologically significant for any alveolar or bronchial tissues, such as basal cells, club cells, ciliated cells, and goblet cells. For our research, we performed NGS analysis on collected SRA data from their studies, which is also available in NCBI as open resource. And we conducted a study about the differential gene expressions using both human reference genomes GRCH37 and GRCH38. These are the SRA data which we collected from NCBI, and we divided them into two groups. In the first probe set, we grouped A549 cell lines, human bronchial epithelial cells, and human bronchial organoid to study about the similarity between the cell types. In the second probe, we grouped control human bronchial organoids which were not infected, SARS coronavirus 2 contaminated human bronchial organoids, and SARS coronavirus contaminated and with chemostat treated human bronchial organoids. Coming next to our problem statements, so we wanted to study about the impact of different reference genomes GRCH37 and 38 throughout our analysis. A differential gene expression studies among the control, infected and treated human bronchial organoids. A control test to analyze contamination of our probe 2. As mentioned, different pipelines of transcriptomic analysis were performed using both human reference genomes separately, so GRCH37 and GRCH38 for each sets of probes. Then the gene expression table were quantile normalized. Then a principal component analysis was performed to study the gene expression similarity within the groups in each probe set. The differential gene expression analysis was performed within each type of sample in group 2. In probe 1, it was only performed between human bronchial epithelial cells and human bronchial organoids. Finally, we plotted the differential gene expressions on the available KEC pathway diagrams to precisely investigate the influence of the reference genomes. As a metagenomic part of our study, a metagenomic microbiome analysis pipeline was performed using all viruses as a reference genome. Then the read count of different virus genome were obtained from the pipeline output file, and the read of all the coronavirus genome were extracted out of the table and annotated with the help of Excel, Tableau, and NCBI viral genome list. We performed a lot of pipelines, but only important results out of our investigation will be discussed here. From our analysis report of probe set 1, we were able to get results from different transcriptomic pipelines, and the results were identical in both GRCH38 and GRCH37. We could observe a clear grouping in our principal component analysis report as well. At the same time, organoids are more alike to human bronchial epithelial cells comparing with A549 cell lines. In our differential gene expression analysis, we did not observe any significant differences, which was influenced by the reference genome. Dendrogram of our heat maps looks identical in both human reference genomes, and there is neither major difference was observed in volcano plot. Moving on to the KEC pathways, we were able to observe additional differential gene expressions for GRCH38 in steroid hormone biosynthesis pathway and hematopoietic cell lineage pathway. As next, moving into the second probe, we had issue performing transcriptomic pipelines with the reference genome GRCH38, although with the GRCH37 we were able to obtain results. We were not able to conclude anything about the quality of our probe or pipeline errors, since we were able to get results for the GRCH37. Moreover, we had results for probe 1 for both GRCH37 and 38. Then a transcriptomic analysis pipeline using sailfish method was performed without any pre-processing to get the read table without any errors. In our principal component analysis, we were not able to observe a clear grouping as we observed in probe 1. Then we decided to do a batch correction. However, we were convinced to continue without batch correction, as the scatter plot after batch correction looks so artificial. We could see a clear separation between control organoids and contaminated organoids, although there is no significant separation observed between infected and treated organoids. We proceeded with the differential gene expression analysis. As a differential gene expression analysis could be performed among two sets of samples, we performed the analysis in 3 further groups. 1. Control and infected human bronchial organoids to study the gene expressions while the human bronchial organoid is infected. Secondly, the control and treated human bronchial organoids to see whether we could apply it as a method to study the side effect of our drug. Thirdly, we analyzed the infected and treated human bronchial organoids in order to see the significant gene expression throughout the treatment. From our differential gene expression analysis between control and infected human bronchial organoids, we could not witness any vital influence of different version of human reference genome. The dendrogram looks similar in both heat maps, only some of the genes were differently arranged on their rows. Volcano plot is also comparable. However, there is a notable difference in gene expressions between control and infected organoids. The differential gene expression analysis between control and treated human bronchial organoids also did not show any notable influences of the reference genome. The dendrogram of the heat maps look the same. As a previous result, there is a notable difference in gene expression between control and treated organoids. But the differential gene expression analysis between infected and treated human bronchial organoids does show us a difference in gene expressions, which was influenced by a reference genome. The dendrograms shows the different groupings depend on different genome, and there is a notable difference seen in volcano plots as well. Moving on to the differential gene expression analysis on KIK pathways, we did not observe any significant difference among the control and infected organoids, or control or treated organoids. But there was an additional pathway observed in differential expression analysis on KIK pathways for reference genome GRCH38, which shows the down expression in pentose and gluconorate interconversions pathway on treated bronchial organoids. It could be significant for the biochemical or physiological signaling pathway investigation of bronchial organoids as an effect of chemostat treatment. As there was significant difference noted in the heat maps and the volcano plots of control organoids and the treated organoids, the remarkable difference was expected in their KIK pathway analysis too. As mentioned in the slide, we were able to see completely different pathways analysis diagrams depend on the human reference genomes. Even though there are 6 KIK pathways that were common among the both reference genomes, there were some unique pathways observed for each reference genome, which probably important evidence for signaling pathway studies of this experiment. Those are the important data and visualizations from our transcriptomics part of this study. Okay, let's move on to the metagenomic part of our research. Although there isn't any clear evidence to prove that the control organoids were not contaminated, we could observe there's a difference in their counts. For example, the infected organoids shows some more counts than the control organoids. Additionally, we could observe the absence of SARS coronavirus 1, which is HKU1 in control and treated samples, which was but found in infected organoids. It is an interesting topic to be analyzed furthermore. As we looked into the significant genes for the basal cell, club cells, ciliated cells, and goblet cells, we could recognize that the TMPRSS2, ACE2, CC10, FGF1, IFN1 genes are very unique for the human bronchial organoids. As the research is all about TMPRSS2 inhibitor, it is evident that the organoids are the best study model among them for this research. About the second table, we could see the difference in TMPRSS2 expressions comparing with infected organoids. 2/3 of the treated organoids show less TMPRSS expressions, which is almost similar to the expressions level of the control organoids. Although chemostat shows the positive response as TMP-RSS2 inhibitor, we cannot conclude it as a successful medicine for SARS coronavirus 2 because the experiment was conducted among 3 sets of organoids. Finally, let me come to the conclusions and finding from our study. There are some differences noted in our differential gene expression analysis, which was influenced by the reference genome. Especially, the differences were significant among organoids, but we were not able to choose a better version among them. For a better data-driven research, it is recommended to use both reference genome for a wide analysis. Although we face some problem producing a gene read count table for organoids using a reference genome GRCH38 in few pipelines, we were able to investigate the differential gene expression analysis using the sailfish method. So it is also important to try different pipelines methods for a better results. We were able to summarize all the differential expression of significant genes about 33 cake tables, which could be a breakthrough for new biochemical and physiological researches about SARS coronavirus 2 and human bronchial organoids and its signaling pathway studies. Further investigation on this topic may lead us to the invention of new pathways and alternation in the existing pathways, as we noticed theoretically influenza A pathway is additional information for our study, precisely the HSA 05164 cake pathway table. A study comparing a human bronchial organoids and pulmonary tissue biopsy could give us a real breakthrough in the development of human organoids as a drug or vaccine test models. Especially, it could help pharmaceutical industries, scientists, and medicina to avoid unexpected outbreaks during their experiment on living test models. It could also help us to reduce the consumption of time, as the organoids could be manufactured quickly than other living test models. Thank you. Finally, I would like to thank for Elia and PineBio for giving us a interesting program and the opportunity to present our research project. And I would like to thank PineBio educational platform and server platform, RStudio, which helped me in some of the data visualizations, and Research Institute for Microbial Disease, who initiate the core research, which we chosen later on, and NCBI for providing the SRA data, and BioRxiv, the preprint server for biology, and my universities for providing us information on coronavirus, and my former uniclinic, where I could still access my histology preparates, and Nishi for introducing PineBio in Sri Lanka. Thank you.